Namaste children. Today in class 12 biology we are going to learn a new topic, that is biotechnology and its applications. So biotechnology has got many applications in different fields. Okay, so today we are going to learn about the applications of biotechnology in the field of medicine. The first and the foremost thing is we will be learning about how insulin has been prepared. It is prepared by the process called as genetic engineering. So we will be learning about this topic, genetically engineered insulin. Before going into the procedure, we have to know the structure of insulin, how it is prepared and what all different polypeptide chains are there inside this insulin. So let us know about the structure of insulin. In our body, insulin is produced by beta cells of islets of Langerhans, and they are present in the pancreas. We know that pancreas is both an endocrine and exocrine gland. Yes, so this insulin which is prepared in the pancreas is synthesized as an proinsulin. Its inactivated form. Okay, so this proinsulin has got three polypeptide chains. They are A, B and C. So the A polypeptide chain consists of 21 amino acids. B consists of 30 amino acids. The C polypeptide chain acts as a linker, as you can see in this diagram. So this is the A polypeptide chain, this is the B polypeptide chain, and C actually acts as a linker. Yes, it links both A and B. Yes, so here if we draw these polypeptide chains in a linear fashion, then probably we can see that this is A, right? And here you have the sequence of C, and again you have the B polypeptide chains. So this is the linear arrangement of polypeptide chains in insulin. Now, this C chain is called as a linker chain because it actually connects both A and B polypeptide chains. And not only that, it brings both A and B polypeptide chains closer so that they can form disulfide bonds. There are some amino acids which contain sulfur in both the A and B polypeptide chains. So when they come closer, then disulfide bonds are formed between them. Once these disulfide bonds are formed between A and B chain, then there is no requirement of the C polypeptide chain. So it disappears, and then the molecule which we have, the substance which we have, is now called as the active insulin. So this is the structure of the active insulin with its disulfide bonds together. Now we will see how insulin has been produced by this genetic engineering process. Scientists have observed that there is no requirement of the C chain in the active insulin. So they have found out that this A polypeptide chain is being coded by A gene and B polypeptide chain is being coded by the B gene, and henceforth even the C polypeptide chain. But in the active insulin, the C chain is not required. So here they have exploited this quality of the active insulin and isolated the gene responsible for only A polypeptide chain and gene responsible for only B polypeptide chain. They have actually taken the gene for A polypeptide chain and then they have transcribed that and inserted in the prokaryotic cell. And the same thing happens with the B polypeptide chain also. Now basically when we come to the actual protein synthesis, we see that if a gene has to be transcribed, then it is the first hnRNA which is formed. This hnRNA consists of both the coding and the non-coding sequences like the exons and the introns. So by the use of endonucleases and by the process of splicing, these exons and introns are separated and the introns, that is the non-coding sequences, are removed. And by the help of ligase, the leftover exons are actually joined together to form a mRNA molecule. And then for this mRNA molecule, capping and tailing is done in the 5' and the 3' end of the polypeptide chain, and that is how this mRNA molecule is synthesized. Now, for this process to take place in eukaryotic cells, we have different enzymes that are responsible for splicing and in joining of the exons. These enzymes are absent in case of prokaryotes. Now, they are only present in case of eukaryotes. So, they have directly taken this cDNA and incorporated the cDNA into the plasmid, and which is again incorporated into the E. coli, that is a prokaryote. Okay. So, here this prokaryote will be producing only the A polypeptide chain, as we have inserted the gene for A polypeptide chain only. The same procedure was done even for the B polypeptide chain, and then the cDNA of the gene coding for B polypeptide chain has been inserted into the plasmid, and then the recombinant plasmid has been inserted into the E. coli. Now this E. coli started producing only B polypeptide chain. The isolated polypeptide chains are kept in appropriate conditions so that disulfide bonds are formed between them, and then that leads to the synthesis of the functional insulin. And this genetically engineered functional insulin is called as humulin, and this has been introduced by a pharmaceutical company which is Eli Lilly, and it has been introduced in the year 1983 on July 5th. Now, in the process of preparing this genetically engineered insulin, there are other scientists who have done their work. They are Benting and Best. They have isolated insulin from dog's pancreas. The other scientists are Frederick and Sanger. They gave the sequence, the amino acid sequence of insulin. Now, other than that, Eli Lilly and Ranbaxy, both together, they have introduced an analog to human insulin. That is called as Humalog. That analog of human insulin is called as Humalog. And this Humalog takes less reaction time. It can be activated within a short period of time when compared to Humulin. In the next video, we will be studying about monoclonal antibodies.